work I'm doing with Stephen Brenner at UC Berkeley, and the Prince album refers to the fact that in some ways scientific progress in this field has regressed all the way back to the 1990s. And I'll show you how in a second. So a lot of you probably attended the Cyrus Chothia talk this morning where Stephen talked about some of the work that they'd done in classifying protein structures early on. And so back in the early 90s, that was when they'd first passed the boundary of one structure per day. So at that time, crystallographers were solving around 30 structures per month. Now that 30 per month total has now exceeded 800 structures per month. And so what I'm showing on this graph are the monthly totals in light gray and then a one-year windowed average in black. And you can see that it's steadily increased except for one little blip in 2007. That blip corresponded to the RIKEN Structural Genomics program in Japan being defunded. And at that time, they dumped a bunch of their structures into the protein data bank. And so that caused a big spike. But then on average, the number of structures declined afterwards. But you can see that more or less it's been steadily increasing. So this is indicative of an almost exponential growth rate in the PDB. And so we're now at over 800 structures a month. The last year or so includes a large number of structures that have been deposited into the PDB, but where the sequences have been kept secret. And so in those cases, I actually can't tell much about those, anything about them except for the numbers that are there. And so this analysis really only applies through the beginning of 2014. I should also note that in green, I'm showing the number of residues in the newly deposited structures, and that's on the right axis. And you can see that the number of residues has been steadily catching up, and growing even faster than the number of structures. So that's indicative of the fact that the structures that are being deposited are now larger than ever before. Now, unfortunately, though, the number of new families that are being solved is not increasing in the same way. Now, solving a new family, or solving the first structure of a new family, is very important because it allows the other structures in that family to be modeled. So it also allows, occasionally, the inference of ancient homology that was not appreciated until structures from that family were solved. So for example, on the basis of sequence, you might think that two different families are not related, but after you solve a structure from both of them, you can see on the basis of structure that they really are from an ancient homologous ancestor, or they're homologous and evolved from a common ancestor. So what I'm showing in this graph is the number of new families being solved per year. So back in the early 90s, when there were 30 structures a month being solved, about 5 of those were from a new family. And that number steadily grew. Around the late 90s, people started doing structural genomics, and that number grew, partly as a result of structural genomics, to a high of about 40 families per month in the mid-2000s. But then since 2007, it's really fallen off. And today, or actually around 2014, it's at the lowest level that it's been since 1996. So I'd like to provide some context for these numbers by showing you that, again, 2007 was when the RIKEN Structural Genomics program shut down. That was an important milestone. In the United States, a lot of the structural genomics funding was provided by this PSI, which is the Protein Structure Initiative, which was conducted in 3 different phases. In PSI 1 and 2, there was a lot of emphasis on solving the structures from new protein families. But there was a lot of backlash as to whether that was a worthy scientific goal. And so in PSI 3, they basically switched the goal to solving the structures of medically relevant proteins, or proteins of known medical importance. And so that was called PSI biology. Okay, and so in the late 2000s, before 2010, the structural genomics centers, knew what the goal of the 3rd phase was going to be, and so they started switching their target selection to accommodate that new goal. By 2010, in the 2010 to 2015 period, they had completely transitioned to PSI biology, and so the production of new families fell off even further. Now, this is in contrast to, as Stephen showed in his talk earlier today, in the mid-2000s, structural genomics were providing almost half of the new families that were being solved. And so you can imagine that today, since the structural genomics production is essentially zero, basically all of the structures are provided by structural biologists, and so, or all the new families are provided by structural biologists. And so that had already been declining starting in 2000, and so that's why we're down to only 17 families a month today. That means that only about 2% of newly solved protein structures represent the first structure from a new family. Now, this actually has pretty profound consequences for other aspects of biology as well. You know, this is not just stamp collecting, but it's actually affects medical or human health and medical research. And so one example I'm going to talk about is the analysis of rare genome variants and how that's been made more difficult by this lack of new families. And so this diagram up here basically represents sort of a funnel. When you sequence the exome or you sequence the genome of a person with a rare genetic disorder, you'll find hundreds of thousands or even millions of SNPs. And what you want to do is figure out which of those SNPs might actually be relevant to the disease that the person has. And so that filtering can be represented as sort of a funnel where you try to figure out, you get it down to a small number. And having the structures of more families is critical to that, as I'll show in a minute. The second thing, which I'd like to talk about, is curation of structural databases such as SCOP or CATH or ECOD. Basically, the low number of new families is actually beneficial to structure classification because it means that a partial manual strategy is actually feasible, even if you don't have someone who can memorize 100,000 PDB structures the way Alexei Mirzin can. So first thing I'll talk about is screening variants. So structure is a very helpful criteria that genetic variant analysis people can use in determining whether a mutation actually has a deleterious phenotype or not. And so in this example, two of these genetic variations from a list of SNPs might be mapped onto a crystal structure or a model for that family. And you could use the crystal structure to determine whether the variant is likely to cause a phenotype. Generally, if it's in the interior, it's bad. And if it's on the surface, unless it's in an important active site, for example, it's usually not bad. However, these last two mutations could be in a family that doesn't have a known structure, and so you wouldn't be able to use that as a filtering criteria. Now, the thing I'd like to emphasize is that sequencing has made more and more families large over time. As sequencing's gotten cheap, the number of families, say, that have 1,000 or 100 members has gone up to almost 10,000. Even the number of families with 1,000 members has gone up to several thousand. But because structural genomics ended and the number of new families that are being structurally characterized hasn't kept up with sequencing, the fraction of those families that are large that has a known structure has fallen from 70% 10 years ago down to about 50% today. And the same is true for even larger families. Now, what would have happened if we had continued investment in structural genomics? So this is just one projection of what might have happened had funding continued to be available and the goals of structural genomics stayed the same. We might have had an increase in the number of families that was on the order of the increase we saw from the late 90s to the mid-2000s. And if that increase had continued through today, we'd have solved over 3,000 more families. Now, I think that's a little bit unrealistic to imagine that budgets would have continued to increase even with all the other spending priorities of the governments. So this is maybe a more realistic scenario of what would have happened if we just continued some of the funding and not changed the goals. We would have still solved on the order of 1,200 more families than actually were solved by today. Now, the consequences for coverage of the human genome are here. Right now, with the actual structures that we've solved today, we can cover about 60% of the easy parts, that is the soluble globular parts of the human proteome, can be mapped to one of these families with a known structure. However, if we'd solved only 1,000 more families, we'd be at about 80% coverage if those families were picked in order to maximize human residue coverage of the human proteome. And if we had 3,000 more families, we could get up to 90% coverage. Now, getting to 100% would take on the order of 10,000 more families. I agree that's a little bit unrealistic, but even a little bit of effort would have vastly increased the amount of human coverage we have today. So with that, I'd like to shift gears and talk a little bit about structure classification. So we showed about 10 years ago, Stephen Brenner and I did a study where we showed that in protein structures that didn't have any sequence or any apparent homology to previous structures based on their sequences, we found that about half of those structures represented either a new fold or a new superfamily in SCOP. Now, the other half represented an existing family or an experiment, but basically this showed that if structural biologists chose to target structures without any sequence similarity to existing structures, you could get a lot of novelty out of the things they solved. On the other hand, if structural biologists just pick targets the way they've always picked targets, based on what they're interested in, the percentage that represent a new fold or new superfamily in SCOP is only about 2 to 3%. And so most of what they choose to work on, at least over 70% of what they choose to work on, is simply more experiments on a previously characterized protein. That's not to say that those experiments aren't novel in other respects. For example, they might be solving the structure with a new ligand. They might be solving a co-crystal structure with several proteins. But in terms of solving new families, they weren't really making much progress on their own until they were paid to do so by structural genomics programs. Now, this just shows that the timeline of old SCOP version 1 releases, and these are the new releases we've done of SCOPE, which is our extension of the classic SCOP hierarchy. And so in the last few years, we've released 5 stable versions of SCOPE that have increased our coverage of the number of PDB structures. SCOP had about 38,000 PDB structures classified at last date. Now we have over 65,000. And you see we've been able to keep up pretty much with classifying about 2/3 of the PDB. Because so many of these structures are not very novel, it's almost trivial to classify them using automated algorithms with the same accuracy as manual curation. Now, because though we haven't classified any really novel stuff until recently, in 2014, we started manual curation again. And so in the last 2 releases of SCOPE, we've manually classified about 60 or so new superfamilies into the hierarchy. And then we're using our automated methods to extend that to other protein structures. And just as we predicted, based on the slide I showed you a couple of times ago, these new Pfam families that we've been manually classifying into SCOPE have actually resulted in a number of new folds and new superfamilies. In fact, these numbers are even a little bit better than the numbers we had projected, because Pfam is a more sensitive way of searching for remote homologs. And so if something is a new Pfam family, it's even more likely to be a new SCOP superfamily or fold when it's classified in SCOPE. And so I'd like to talk a little bit about the pipeline we're using to classify structures. As I said before, SCOP has about 38,000 structures out of 110,000 in the PDB. About 70,000 of those are now in SCOPE based on this automated pipeline that, again, it classifies all the trivial structures with no errors. Now, about another 20,000 are clearly members of existing SCOP superfamilies, but the homology in those cases is a little bit more remote, and so we can't necessarily accurately assign the domain boundaries with a fully automated process. And so what we do is we predict the boundaries using existing tools and then we're putting them into a queue to have a human validate or correct the boundaries before we put them into the database. And then finally, if we look at the unclassified stuff that's in Pfam families that have been structurally characterized but are still waiting to be manually curated, there's over 2,000 of those families. And so I expect that when we manually curate those families, again, about half of those families will result in new folds or new superfamilies. And so the idea that we're running out of structural novelty is really incorrect. There's plenty of stuff that's actually already been solved and has yet to be classified into SCOP or into CATH for that matter. And there's also plenty that remains to be solved because structural genomics has ended, and so the rate of characterization of new families is now very low. And so finally, there are some protein structures that are being solved that are not in Pfam, and those are in this pipeline too. They're just lower priority than the Pfam families. And with that, I'd like to acknowledge my co-authors on the SCOP project, as well as on Astral and on SCOP. I have a poster that I presented if anybody wants copies, and I'd like to thank the NIH for funding, and thanks for your attention.
We have time for a couple of questions. Alex?
So thanks for a very interesting presentation, and thanks for the Pfam advertising, much appreciated. So solving structures, I kind of feel that there are two types of structures. There are the globular proteins, and then we have the membrane proteins, and the properties and challenge of solving those are quite difficult. Right. When you made those estimates of the 80%, 90%, you weren't including the membrane proteins. So could you say something about how things might look there?
Right. Basically, I've also calculated the number of residues that are in either membrane proteins or intrinsically disordered proteins, or parts of proteins that are not terribly interesting, like coiled coil. I mean, well, I guess it's interesting, but not from a structural perspective usually. And so that would add about 20% to all of those bars that I showed. But basically what I was showing was that the parts of proteins that are easiest to solve, we've only solved 60% to date. Obviously we can solve a lot of membrane proteins too. It's just a little bit slower and more expensive to do so.
I have two questions. The first, can you speak to the use of coevolution methods for structure prediction? How much more coverage would they give you if one were to use them? And the second question, just to clarify, the 1/3 that's not yet in SCOP, that's because they just haven't gone through your manual curation pipeline?
Exactly, right. For a while, we were focusing on developing reliable automated methods that could handle 2/3 of, or actually they handle about half of the new structures that are coming out with the same accuracy as manual curation. We wanted to increase the number of structures that were in the classic SCOP without compromising the quality of the database, because it's been built up over 20 years by Alexei Mirzin to have really high quality. I estimated that his error rate is in the order of 1 in 10,000. So it's a very high quality database, and so we had to very carefully benchmark our methods to make sure we didn't introduce more errors. And so that's why we're being pretty conservative about what we're putting in. There's about 20,000 or 25,000 structures that are waiting to go in, but I'll need to manually check them before we add them. And the other question about coevolution, what was the question there?
Yeah, how much more coverage would that give you if one were to use coevolution methods? Because they're only applicable to large protein families.
Right. So I'm not really considering predicted structures here. I mean, obviously more families could be modeled, maybe using David Baker's methods or what have you, but I'm talking about ones that there is an actual structure for at least one member of the family.
Okay, thank you very much, John-Marc.